Namaskar. Today I am going to deliver a lecture on chromosome, especially Euchromatin and Heterochromatin. What are previously in a part 1 I have discussed about what is chromatin, what is the structure of chromatin, and today we will discuss about what are the different types of chromatin. So here very important is that chromatin can be divided into two parts, euchromatin and heterochromatin. What are the euchromatins and what are the type of heterochromatins? If we will see a chromatin structure, it is a chromosome, and inside the chromosome there is a chromatin. Previously discussed, I have discussed it, and it's a chromatin, and this chromatin is highly coiled. It's present in a highly coiled state inside the chromosome. So this is the chromatin, and if we will see that the chromatin is not uniform or uniformly packed with the histone. Sometimes chromatin and histones, chromatin that is DNA and histones are highly packed, sometimes they are loosely packed, or during the cell cycle sometimes chromatin remains in an uncoiled state. Here is a chromosome structure where if we see that this is a chromosome, and correspondingly this one is a chromatin present inside the chromosome, and so some region of the chromatin, here the telomere region, in the telomere region, in the centromeric region and the telomere region, two telomere and the centromeric region, chromatin will be compactly packed and they remain in a packed condition. These are the heterochromatic region. So I have divided here, and rest of the areas are less coiled and highly active, uncoiled structure and highly active, especially in the interphase stage of the cell cycle, and these uncoiled parts are euchromatin, and these euchromatins are highly active, especially during the interphase stage when proteins are synthesizing. So euchromatin regions are actively participating, euchromatin regions actively participate in protein synthesis and high gene concentration, that is genes are present in a high number in euchromatic regions. In heterochromatic regions genes are not present and they remain uncoiled state, they remain in a coiled and condensed state, and they are not taking part in a protein synthesis or in active gene activity. So these three regions are very important in a heterochromatic region. So chromatin can be divided into two parts, euchromatin and heterochromatin. Euchromatins are uncoiled part of the chromosome, that is in an interphase stage, that is during active cell cycle stage. Now when proteins are synthesizing, euchromatin regions are uncoiled. They are working, they form mRNA and protein. They actively transcribe, and transcription continues in that region. But these heterochromatic regions, transcription is hindered, a transcription not occurs. So in a heterochromatic region can again be divided into two parts, constitutive and facultative heterochromatic regions. What are the constitutive and what are the facultative heterochromatic regions? Constitutive heterochromatic regions are those regions in a chromosome which are permanently remaining heterochromatic stage, that is permanently in a coiled stage or permanently in a coiled condition. These are constitutive heterochromatic region, that is they in any stage of the cell cycle they remain in coiled stage and not actively participate in a protein synthesis. So these are the constitutive heterochromatic region. The centromeric region is a constitutive heterochromatic region. No genes present in centromere, in telomere, and in most part of the male Y chromosome. So these are the constitutive heterochromatic region. And what is the facultative heterochromatic region? In our chromosomes, some part actively participates, that are the euchromatic regions, and some parts, that is the heterochromatic, among the heterochromatins all heterochromatic are not constitutively heterochromatic. Some heterochromatic are periodically heterochromatic, and periodically they are in an uncoiled state. Sometimes they coil state, but sometimes in uncoiled state. Very popular example is the one of the 2X chromosome of females. We know that the female cell contain 2X chromosome, but both the 2X chromosomes are not actively participates in protein synthesis. One of the X chromosome remain coiled stage or remain in a heterochromatic stage during the interphase stage. So if we will see the mammalian cell in an interphase stage and we will stain it, we will see that a spot, a deep spot, and that spot is nothing but one of the 2X chromosomes, and that remain X chromosome. One X chromosome is active, but another X chromosome remain in an active stage or coiled stage. It is called Barr body. And we all know that what is the Barr body actually. One of the 2X chromosomes exhibit facultative heterochromatic region. This Barr body, one of the 2X chromosome, remain inactive during the interphase stage. That is, when cell is functioning, all chromosomes are functioning, it is remaining non-functional state. And during the metaphase stage, it becomes uncoiled. So this is a facultative heterochromatic region. That is, one of the 2X chromosome is facultative heterochromatic, and it is a Lyons hypothesis also. That is, not both the X chromosome is active in females. One of the X chromosome is inactive during the interphase stage. Now, so what are the difference? What are the features of euchromatin and heterochromatin? Features of euchromatin: during the interphase stage of the cell cycle, that is G1, S and G2, all euchromatins will take light stain. If we stain the cell or chromosome, they will take little stain during the interphase stage. All chromosome, because they remain in uncoiled state. They remain in uncoiled state and functional state, and translation occurs. So they remain in an uncoiled state and light staining occurs. But heterochromatic region, heterochromatins in that particular time will take a deep stain. That is, during the interphase stage, heterochromatin will take deep stain. That is, they are not working. So why they remain in coiled condition and take a larger stain and greater stain, high stain density. However, in an interphase stage, active transcription occurs in case of euchromatins. In case of eukaryotic chromosome, that is during the G1, S and G2, these three phases, the active transcription phase, they will actively transcribe and produce mRNA for the synthesis of protein. But they are transcriptionally inactive. Heterochromatic regions are transcriptionally inactive and not taking part in protein synthesis. And during the replication, that is during the formation from one chromosome to another chromosome, the euchromatic regions are replication occurs very early in that euchromatic regions. Rather, replication occurs late in heterochromatic regions. Euchromatin is found both in prokaryotes and eukaryotes, but heterochromatids are only found in eukaryotic regions. One important thing is that these parts of the chromosome remain uncoiled condition, These three parts remain coiled condition, that is the heterochromatic regions, but they uncoil only during the DNA replication. During the DNA replication, they all uncoil, but during the working time, they remain coiled conditions, the heterochromatic regions. And one of the important thing is that housekeeping genes are those genes which require active transcription in all times, in all 24 hours, 65 days. In that case, these are all euchromatins. So housekeeping genes are all euchromatins. They should be euchromatin, because without euchromats, if they will be constitutive or facultative heterochromatin, then housekeeping gene will lose their function. So today I am discussing about chromatin. What are the different types of chromatin, euchromatin and heterochromatin, that I have discussed, and the important thing is that what are the different